Hi, so yeah, first of all, just to echo what Ellie has said, thank you so much to UK Spine for actually funding this proof of concept work that we're going to do. And thanks so much for giving me the invitation to talk about it today. So yeah, I am Lorna Fitzpatrick and I am a lead scientist in cell and molecular engineering at MDC. And I will talk you through today our proof of concept project, which is actually a collaboration between MDC and Dr. Nicholas Rattray at the University of Strathclyde. And the project is focused around epigenetics and aging and can modulation increase lifespan? So actually this proof of concept work is a follow-on from a previously UK Spine funded project that we started in about 2020, myself, Graham Wilkinson at MDC and Panagis Philippakopoulos at the University of Oxford. So first I'll just go into a bit of background about that project, the results we got from that, and then hopefully that'll help you to understand where we got to with this proof of concept project today. So our first study is focused around two papers that came out of the Kai group in Shanghai. And what they found was that they saw a decrease in BAS1, which is a dopa decarboxylase, in aging in a C. elegans model. And what they found was when they saw this drop off in dopa decarboxylase, they also saw a drop off in a couple of neurotransmitters, dopamine and serotonin. This drop in the neurotransmitters then led to a deterioration of the behavior of the worms, whereby they saw changes in pharyngeal pumping and male mating. So then in 2020, the same group performed a follow-on study. So they did an RNAi genome-wide screen looking for BAS1 regulators. And of those that they identified, they focused on two epigenetic genes called SET6 and BAS2. So SET6 and its human ortholog EHMT1, they're both methyltransferases. And then BAS2, there's actually not a lot known about the function of BAS2, but it's thought to be a chromatin remodeler. So what they found was in aging C. elegans, they see this increase in SET6 and BAS2, and this increase then accelerated the aging process by modulating mitochondrial genes and thus having an effect on mitochondrial function. So what they found was when they inhibited both of these genes, they saw improved function in the C. elegans, improved mitochondrial function, an increase in BAS1, increase in dopamine production, and so on. So that's what our project was kind of focused on in the beginning based on these two studies. So as I said, these are both epigenetic targets that we're working on. And actually the relationship between epigenetics and aging is very complicated, but in a very loose term, in young individuals, cells within the same population generally have the same transcriptome due to having the same epigenetic profile. And what happens is as the cells age, they become exposed to different endogenous and exogenous stimuli that then can cause problems. And what you get is abnormal chromatin structure, whereby you get inclusion of different histone variants, you get unusual histone modifications, and then changes in DNA methylation, which then ultimately change the whole transcriptome of the cells, and you get this transcriptional drift in older cells. So in terms of histone methylation, this can either be activating or repressive. And in the Kai study, what they found was when they saw this increase in BAS2 and SET6, they saw an increase in histone 3 or H3 lysine 9K9 trimethylation. And when they saw this increase in H3K9 trimethylation, what they saw then was a reduction in mitochondrial function due to a repression of mitochondrial genes. So then inversely, when they inhibit, they see an increase in mitochondrial function and lifespan. So this is where we come in. So there are a number of tool compounds out there that can target both EHMT1, which as I said is the SET6 human ortholog, and targeting BAS2 as well. So we looked at these two compounds, A366 and GSK2801. Both were developed in partnership with the SGC. And firstly, A366, it's a potent inhibitor of both EHMT1 and its paralog EHMT2. And when you treat a number of different cell lines with this inhibitor, what you see is global reduction in H3K9 mono, di, and trimethylation. And then GSK2801, as I said, there's not much known about the function of BAS2. However, this compound is known to selectively target both BAS2A and BAS2B. So our plan was to understand if either of these compounds had the potential to increase longevity in an in vitro human neural cell model. So we set out 2 work packages. The first one was in a neuronal cell line, which is called SHSY5Y, and it's a neuroblastoma cell line often used in neurodegeneration studies. And the reason why we chose this line is because, according to the RNA Atlas, it actually expresses quite high levels of dopa decarboxylase, which is the human ortholog to BAS1. And these cells are known to have some biomarkers of dopaminergic neurons. So the goal for this work package, firstly, was to look at target engagement. We wanted to use compounds and do genetic ablation via siRNA to see if we could see any phenotypic effect. So in the cell line, we wanted to have a look at global methylation, ATP production, mitochondrial function, etc. And then when we were happy with the dosing that we had from the work package 1, we wanted to then move into a more relevant cell type. So we wanted to look at similar things, global methylation and ATP production in primary mouse neurons. Okay, so these are the results from our first work package. And what you can see in the top left-hand corner is a Western blot probing for EHMT1. So what we can clearly see in our conditions treated with control siRNA and BAS2B siRNA, we see nice levels of EHMT1 protein, but then when we knock down with EHMT1 siRNA, we see a loss of that protein. So we're happy to know that, first of all, we actually had target expression in the cell type, because we weren't sure if we were actually going to have target expression in these, and we were happy to know that we could knock it down. Similarly, then on this side, we can see a blot probing for EHMT2. And again, in the BAS2B and the control conditions, we see good levels of target expression. However, when you knock down EHMT2 with EHMT2 siRNA, and when you knock down EHMT1, we see a drop off in EHMT2 protein production. Initially, we were surprised to see this loss of EHMT2 with EHMT1 treatment, but actually, in an endogenous situation, EHMT1 and 2 work together in a heteromeric complex. And it's been shown that in mouse, when you lose EHMT1, you actually do see a loss of EHMT2. So this kind of matched up with what we saw in the literature. In terms of BAS2B, we couldn't find any decent antibodies or that would work in our hands to actually detect protein, BAS2B protein, in these cells. However, we were able to detect transcript, and we were able to knock that down using siRNA. When we treated our cells with compound, we didn't see any change in target expression, which is what we would expect. So then moving on, looking at methylation, what we found, which was quite nice, we saw a dose-dependent decrease in H3K9 monomethylation in response to A366 treatment, so EHMT inhibition. We didn't see any changes in di or trimethylation, which is what the Chi study saw, but yeah, this is what we got. So we were happy enough that we had at least some target engagement here. In terms of GSK, we didn't see any changes in mono, di, or trimethylation. So then we moved into looking at the functional effect. So using a ATP glow assay, we looked at ATP production in our cells in response to compound treatments, and we didn't really see any significant changes. So then we decided to have a look in more depth using the seahorse. So what you can see here in the top right, with increasing doses of A366, we do actually see an increase in oxygen consumption rate and a little bit of an increase in the reserve capacity of the mitochondria. However, we also did see a drop off in extracellular acidification, which would suggest actually there's a little bit of tox with the A366 that we didn't really see when we were looking down the microscope. In terms of GSK2801, we do also see a slight dose-dependent increase in oxygen consumption and a very, very small increase as well in reserve mitochondrial capacity, but not really to a massive extent. So moving on then, at the same time, while these studies were going on, we also did some RNA-seq on cells, 5Y cells treated with both siRNA and compound. And really, obviously, what you can see here on this side is that when you inhibit BAS2B either by siRNA or by GSK, we don't see any major changes at all in the transcriptome, suggesting that perhaps BAS2B doesn't really play a role in regulation of these cells. However, we did see quite nice results when we inhibited EHMT. What we saw was around a 70%, just above 70% correlation between the gene expression in EHMT1 and EHMT2 knockdown cells. We also saw around just over 70% correlation between EHMT1 and A366 compound, and then slightly less, but we still saw a really nice correlation between EHMT2 siRNA knockdown and A366. So actually it was Panagas who did all of this bioinformatic work, and then what he went on to do next was look at gene ontology analysis and gene set enrichment analysis as well. And what he found that there was an enrichment for a number of genes and a number of pathways, such as adhesion molecules, receptor activity, translation. A few genes came up that were associated with risk of developing neurodegeneration, but nothing massively stood out to us in terms of mitochondrial function or dopa decarboxylase activity. However, what he did find was that what he saw was a reduction in the expression of genes associated with midbrain neurosubtypes in response to siRNA treatment and A366, so inhibition of EHMT. And how he figured this out was that basically what he did was he mapped our RNA-seq data onto a data set that was published back in 2016 by this group, Mano. And what they did was they did single-cell seq on samples from mouse and human during the course of development, and they identified a number of different neurosubtypes. So we were able to map that back on, and yeah, we found that we saw an inhibition of developmental genes. So this wasn't too surprising, because actually there is a disease called Kleefstra syndrome, which is caused by deletion or mutations in EHMT1, and patients actually present with neurological problems. So they have developmental delay, intellectual disability, and epilepsy. So I think basically what we kind of concluded from the study was that we were very clearly getting target engagement, but this cell line is probably more reminiscent of a developmental model than an aging model. So that's why then we wanted to move in and do something similar in the mouse neurons, which we thought would be a bit more mature. So what we found in the mouse neurons was that we get quite nice expression of EHMT1 in the nucleus as expected in these. We couldn't, again, detect BAS2B at all. Either that's to do with them not expressing, or to do with the antibodies not being the best. But we did also see, similar to the 5Y, that we saw this dose-dependent decrease in H3K9 monomethylation. But then, unfortunately, we saw a similar result when it comes to ATP production that we saw in the 5Ys. So when we looked at inhibition of GSK and A366 in the primary neurons, we didn't really see any major changes in ATP production. And even then, when we co-cultured with astrocytes, because sometimes astrocytes can help the maturity of the neurons, again, we didn't. So the conclusions of that study is kind of what I've said already. We clearly saw target expression in both of our cell models, and we were able to modulate methylation with EHMT1 perturbation. We saw a really strong correlation between genetic ablation and compound treatment, suggesting good target engagement of our compounds. And yeah, even though the 5Ys express high levels of the dopa decarboxylase, you know, that's still a neuroblastoma cell line isolated originally from a 4-year-old child. So potentially these cells are quite immature. Similarly, mouse primary neurons are also isolated from very young mice. So that's where we came into this proof of concept project, which we're starting now. So the project that we are conducting here between actually Adam Rolt, who's just started at MDC and he's worked with Lynn Cox previously. He's actually going to take over this project from me as he's much better equipped than I am. But this is a collaboration with Nick Rattray in Scotland. So at Strathclyde, Nick has developed a he has developed a model, an aging model in human dermal fibroblasts. And what he has done is he's developed methodology to look at the proteome and the bioenergetics of those cells using mass spectrometry. So what he found is that when he ages cells, he can get a really nice profile. He sees increase in senescence associated secreting profile. He sees increase in cell cycle arrest proteins and oxidative stress and DNA damage. So then finally, this is what the study will look like. So Nick will send, and already has actually sent us samples of different age human dermal fibroblasts. And Adam has already got these growing in culture. And the idea is to grow these cells in the presence of our inhibitors and see, do our inhibitors have any effect on senescence. So Adam will set up a number of protocols, which Nick is sharing with him, looking at, or that he's actually developed as well previously, looking at beta-gal, EDU, so cell cycling. And then the other thing that Adam will do, which would have been nice to have done previously, is actually he will establish his own model of senescence using chemical induction via mitomycin C treatment. So then ultimately at the end, what will happen is that Adam will ship samples then to Nick, and Nick will be able to do proteomic and bioenergetic analysis on those samples. And then we'll get a really good idea to see how our targets are having an impact in this particular model. So then finally, our overall goals and output for this project are, firstly, we're really keen to develop this integrated proteome metabolic model in response to target inhibition. But then really importantly, what we want to see is if we can validate these targets for progression to the next stage of the drug discovery pipeline that UK Spine is putting together. And we've clearly not tested our compounds in the correct model. So hopefully this will give us a better idea of whether these are targets worth progressing. And then of course, as Janet was talking about earlier on, we're really pleased that now Strathclyde is in the UK Spine Network, and he's been really keen to share protocols, reagents with us and us back with him. So expanding that network. And then finally, Ellie just gave a really lovely talk looking at DSP in young and older mice, and actually has included both EHMT1 and BAS2B in her panel. So actually we'll be able to get an idea of expression of these particular proteins in all of the different organs in the young and old mice. So that's all. Thanks for listening. I want to say a particular thank you to everybody at Catapult, particularly Mary Challoner, who did a lot of the work alongside Panagus. And yeah, happy to take any questions.
So I think we've got time for one question now, but we might have time for some more at the end. So do I have one question now? One at the back up there. Pip right up the stairs. Probably good for her healthy longevity.
Thank you. Those SGC compounds, do we know the potency at the mouse proteins? Do you think that could be the issue, or the other issue is that publication 3 years ago is just wrong?
Yeah, so yeah, that could be the issue, and I think that's something that we need to look at. I think I think we're happy enough that we do definitely see target engagement. I think when Adam moves on looking at the HDFs, I mean, we didn't actually have a baseline level of mono, di, or trimethylation in the beginning. So hopefully now if we actually look and see, is there an increase in trimethylation in an aged model, maybe then our targets might be more relevant. So I'm not sure if it is, but we'll have to find out really in this study. So I'm not sure if it is a problem with the compounds or the original study, or we're just not looking in the right place or haven't so far. So hopefully there are things that we can figure out as we move forward.
Thank you very much.
Thank you.